पण घेऊ शकतो का तसं? प्ले करा ना.
ते काय म्हणतात ह्या म्हणतात ना आधी PPT.
लाईव्ह गेलोय का आपण? Hello everybody, welcome to another session of YouTube Live Standard 9 Science and Technology Part 2. We are going to continue with lesson number 17, Introduction to Biotechnology, right? I hope you are all in good health and have a good life. We are going to continue with this chapter, and I'm Mrs. Priya Mohorkar presenting to you this chapter. We are going to continue with the session 3 of the YouTube Lives. I'm going to present this chapter from SV Union High School and Junior College, Pune, Maharashtra, India. Let us start with this chapter, lesson number 17, Introduction to Biotechnology. Now, last two sessions, we have learned the subtopics under this chapter, which are the tissues, like the animal tissues and plant tissues, isn't it? Today, we are going to continue this chapter with a new subtopic, the tissue culture. Okay? So, but before that, let us recall whatever we have learned in the last two sessions. Okay? A quick look to the initial part of this chapter. Okay, when we look around, we know that we are surrounded by different things, both living as well as non-living things. The non-living things are those which do not perform any work or function on their own. Like example, just if you take a building, you know that the building is made up of bricks, cement and concrete, iron rods or bars, okay, as the basic structures. The same way, living things which perform lots of work and function on their own, like growth, respiration, nutrition, excretion, reproduction, etc. For example, if we take ourselves, our own selves, man, we are all made up of basic structural and functional units called cells, and along with the cells, we have fluids in our body and the skeleton that form the major part of our body. So living things and non-living things are both found, but we are going to talk about the living things, which are basically made up of cells. Okay, but before that, let us just see what is the hierarchy of organization if we consider the non-living things, right? If we take a book, we saw a textbook, right, last time. The textbook is made up of very small, the smallest part or the smallest structure of the textbook are the letters of the alphabet of that language, right? When these letters of the alphabet come together, they form words. And when words come together, they form sentences. Sentences are arranged in various paragraphs of different lessons, and all these lessons are found in a textbook, right? Okay, the same way, so even though a book is a non-living thing, it is still organized, right? Let us see how the organization of organism is hierarchied. Now, the hierarchy of organization of organism, if we consider, we know that we all are made up of, that is, organisms are made up of basic structural and functional unit called cell, isn't it? Right, you have already studied about the structure and function of cell. Now, these cells are very, very important, as they are going to form the structure as well as they are going to function for the whole organism. But how are they going to do it? They actually come together if they are similar in structure and function and they are from the same origin, then they form a group of cells which we term it as tissues. Now that is a tissue. These tissues perform different a specific function. Now different cells, when they group together, they modify themselves according to the function. Okay, now when they modify themselves means they undergo certain changes, maybe in the structure, maybe in the size, shape, and they are going to form together form the tissues for a specific function, to perform a specific function, right? So cells together are grouped together. The cells which are similar in structure and function as well as from the same origin, they are going to form a tissue to perform a specific function. Now, when these tissues are grouped together, they form what are called as organs. Organs like the liver and the stomach of the digestive system, which we have already seen, isn't it? Now, digestive system, what is that? It's an organ system. When organs are arranged together sequentially to perform a specific life processes, then it is called as an organ system. Like here you see the digestive system, isn't it? Now that such systems are found inside a body cavity. They are arranged and they synchronize together, doing their own function as well as helping out others, where you can see the division of labor, division of work amongst these organ systems, and thus the organ systems, different organ systems together form an organism. You can see here different organ systems like the respiratory system, the skeletal system, the muscular system, the digestive system, the circulatory system, and so on, and other all these different organ systems together form an organized organism. Okay, that is the hierarchy of organization of organism. So these organisms may be unicellular or multicellular. Unicellular organisms are single-celled organisms. That is, all the functions are performed by the small organs called as organelles of the cell. For example, you find amoeba, paramecium, all these are unicellular organisms, right? Whereas multicellular organisms are many-celled organisms, they are complex organisms, and all the functions are carried out by group of cells which are organized as a tissue. The tissues, you know, now are organized as organs arranged in an organ system inside the body, which we call it as a organism. Now, if we consider plants, animals, and human beings, we all are multicellular. Right. So we are going to talk about in this chapter about tissues and different types of tissues, both animal and plants. Both these subtopics are being covered in the last 2 live sessions, right? This is the third, and we are just going to revise whatever we studied about tissues. Okay. Let us start. Now, tissue is actually a group of cells having the same origin, same structure, and perform same function. All the functions of the body occur with full efficiency due to specific organization of cells and their division of work. Okay, we call that as division of labor, right? Now, there are different types of tissues, but basically if you see, the tissues are of only 2 types. One is the simple tissue, the other one is the complex tissue. You know, simple tissues are made up of only 1 type of cells, whereas complex tissues are made up of, yes, more than 1 type of cells, right? Okay, now let us see the animal tissues. We now know that animal tissues are made up of animal cells, and we also learnt about 4 different types of animal tissues, which are the epithelial tissue, which are the simple animal tissues, the connective tissues, muscular tissues, and nervous tissue. All these 3 are complex type of tissues. Okay, let us just have a look of various types of epithelial tissues which we studied. the squamous epithelium, the stratified epithelium, the glandular epithelium, the columnar epithelium, the ciliated epithelium, and the cuboidal epithelium. Generally, epithelial cells, tissues uh, are found on the surface, whether it might be an outer surface or inner surface of any organ or the organism, right? So these are the different types of basic epithelial tissues that you have to know, you have to study. Please see to it where they are located and study the structure and function of these tissues, which we have already discussed in detail. Now, in the next type is the connective tissue that connects various organs of the body. We have learned them already. The different types of tissues, let us just revise: the blood tissue, the lymph, areolar tissue, the adipose tissue, the cartilage, the bones, the tendons, and the ligaments. Now these are actually the connective tissues that connect every organ and every part of our body. Now, the next type was the muscular tissues, that is the muscles which help in locomotion, that is movement, and to move from one place to other place is being determined by these muscles. Three main types of three types of muscles, which are striated muscles. Striated muscles, you know that they are also called as voluntary muscles, whereas the non-striated muscles are called as involuntary muscles, and the cardiac muscles, which are the heart muscles, that is, they are found mainly in the heart. So these are the three different types of muscular tissues found in animals. Whereas we also saw the nervous tissues, which help to us to respond to the stimuli surrounding us from the surrounding environment. So there are two different types of nervous tissue. One is a neuron that helps to respond the stimulus by transferring the stimulus from one neuron to the other neuron, and they form a part of the brain, the central nervous system, the peripheral nervous system, and the autonomous nervous system. Whereas there are supportive cells to these neurons, which are called as the glial cells or the neuroglia. Okay, there are different types of neuroglia that helps in supporting the conduction of stimuli through the neurons. Okay, right. So this is the summary of the animal tissues, which we just saw. Let us see what we learnt about the plants, right? You all know plants. They are lush, green, beautiful, large, sedentary organisms. That is, they are fixed with the help of part of the plant that is called root system, and these roots make the plants fixed. Most of the plants, higher plants, are fixed and sedentary, right? Let us see the hierarchy of organization in plants. What are these plants made up of, and how are they organized? They are also living things, living organisms, and that is why they are made up of the basic structural and functional unit called the cell. But here you now know that plants have plant cell with slight differentiation from the animal cells, right? Now, as they are fixed, most of the cells are designed in such a way that they give mechanical support to the plant. Okay, the plant cell generally consists of the cell wall, which is the outermost rigid covering of the cell. They also contain vacuoles, right? Whereas another very important organelle is the chloroplast, containing green color pigment called as chlorophyll. So these are the major differences between a plant and the animal cells, as animals are motile, that is, they move from one place to other place, and flexible enough, whereas plants are generally sedentary. They are fixed to one place, they do not move from one place to other place. Of course, there are certain exceptions in both, but still generally higher plants are all sedentary. They are made up of cells. Here I will call them as plant cells. Okay, right. So cells, when they are grouped together, of course they are going to form tissues, plant tissues. Okay, so here you can see how the plant tissues are arranged. Now here you can very well see, this is a picture where you can see the walls of the cells. Can you see the wall here, all of you? Now this is the cell wall. The cell wall can be seen through the microscope when we keep a small layer of the epidermal tissue under the microscope, and if you are going to observe them, you can see the cell wall very clearly. Okay, so this part of the cell is going to make the plant as an organism rigid and support mechanically. Cell wall gives mechanical support to the tissues and to the whole plant, right? So different cells, they actually form different types of tissues based on the different functions that they have to perform. So different cells similar cells which are similar in structure and function form tissues, and of course they perform specific functions. Now these tissues are grouped together to form various organs. Now in plants, you know which are the organs of the plant. Can anyone just think over and recall which are the parts of the plant? Of course, you find green color parts, which are the leaves. You find a very hard and thick central strong stem or the trunk in a tree, right? You can also see and you can find the branches of the trees. These so the stem and the branches, of course, all these are various organs. Even the roots, the flowers, the seeds, fruits, all these are various parts of a plant. These parts are organs which are arranged together in a system. Now we call it as organ system, plant organ system. Now which are those systems? For example, you must be knowing that all the roots together which helped the plant to fix in the soil is the root system, made up of roots, different types of roots, right? The root system. And the second type of organ system is the shoot system. Both root system and shoot system combine together, they work together, they are sequentially arranged to form the organism called as plants. Is that clear, all of you? So this is how the plants are organized, and the hierarchy of organization in plants is shown here. Of course, a small plant, when it grows into a large tree, of course, but still has the same hierarchy of organization. Let us see what we learned about the plant tissues in the last session, right? We learned that the plant tissues generally contain three tissue systems. The surface tissue called as epidermis, okay, the ground tissue, and the vascular tissue. These are the three tissue systems which form a plant, which are found in plants. Now, epidermis, which I told you is a surface tissue, contains cells forming the outer surface of the leaves and of the whole plant body, okay, whether it is stem, the root, the branches, the flowers, everywhere you can find this surface tissue, right, called epidermis. Whereas the ground tissue, in the ground tissue, the cells form the inner parts of the stem and the branches of the plant. Now, the third type is the vascular tissues. Vascular tissue, the vasculature, are the primary conducting tissues, conducting components, which are of two types, mainly the xylem and the phloem. The xylem tissues actually conduct water, that is a water conducting system, whereas phloem conducts food, so it is the food conducting system. We also learned that the plant tissues are basically of two types. One are the meristematic tissues and the other are the permanent tissues. But on what basis? Right? On what basis? According to now, according to the organization of the cells, there are two types of plant tissues. The meristematic tissues are actually the basic, very quiet, highly active type of tissues, and they undergo rapid cell division and helps in growth of the plant. Okay, that's why we saw in the last session that plants show unlimited growth, isn't it? That is because of these meristematic tissues. Whereas when the meristematic tissues undergo undergo division and form cells, these cells slowly are going to show some kind of changes in them, modify into different size, shape for different functions, and they form what are called as permanent tissues. But why permanent tissues? Because they are located, their location is permanent after a process called differentiation. That is, when changes occur, when modifications occur according to the function in the cells, and the cells are going to differentiate themselves into various forms or various types, then and are located in a specific place, and that is why when their place is permanent, they are simply termed as permanent tissues. Okay, right. Let us see the types of meristematic tissues now. Now, based on meristematic tissues are classified into three basic types based on their location, where they are exactly placed. Okay, they are apical meristem, intercalary or the intercalary meristem, and the lateral meristem. Apical meristem are generally found at the apex of the plant parts, like the stem tip, the root tip, the leaf tip. Okay, so the tips, the branch tip also. When these tissues are found at the apex or the tip of the plant parts, we term it as apical tissues. Now, when these tissues actually help in vertical growth of the plant. Okay, so the length of the plant increases because of the apical tissues. The intercalary tissues, intercalary are found in meristem, are found at the nodes or just above the node, right, where you can see here just above the node where the leaves arise, the branches arise, or sometimes even the flowers arise in the axil of the leaves, right? So all these structures arise from the intercalary meristem, whereas the lateral meristem is the third type of meristem, which are generally found in the lateral, that is the sides here, this side, sides of the parts of plants, right, like stem and roots. Now here you can see a picture where this lateral meristem tissues are termed as cambium tissue, which are associated with the conducting tissues, xylem and phloem, which are the complex tissues, complex permanent complex tissue. Now, these cambium tissue is meristematic in nature because as they divide, they produce on one side, towards one side they produce the xylem tissues, and towards the other side the phloem tissues, that is the food conducting tissue. Xylem are the water conducting tissue, and they help in increasing the girth of the plant part. Whether it is a stem, then the stem thickness increases because of cambium activity, because of the activity of the cambium tissue, which is a meristematic tissue. Which type? The lateral type of meristematic tissue. Okay, right. So apical meristem, intercalary meristem, and the lateral meristem forms the three different types of meristematic tissues. But remember, based on their location, this classification has been done. Now let us talk about the second type, that is the permanent tissues. Permanent tissues, I told you that they are because the modified cells are permanently located in a particular part of the plant, hence permanent tissues. And there are two basic types of permanent tissues. One is the simple tissue and the other one is the complex tissues, right? you know what are simple tissue and complex tissues now. Simple tissues are those tissues which are made up of only one type of cells, right? Whereas complex tissues are made up of more than one type of cells, right? Complex, isn't it? Okay, is it very simple? Right. The simple and complex tissues are two types of permanent tissues. We saw last class about the permanent tissues and their types also, both simple as well as complex permanent tissues and their types. Let us just recall what we have learned in the last session. The surface tissue, called as the epidermis, is a simple permanent type of tissue, right? You can see the surface tissue is found on surface of the whole plant body, whether it is a root, whether it is stem, branches, leaves, flowers, or fruits. Okay. Now these, we have taken an example of leaf here over here. When we talk about leaf, you can see the leaf blade, which is green in color, right? Now this part of the, if you just peel off the surface of the tissue of the leaf, okay, you can and you can observe this under the microscope. Simple microscope also may do. Compound microscope may do, light microscope may do. Now you can see on the surface you find upper surface, the adaxial side of the leaf, and lower surface, the abaxial side of the leaf. Now both these surfaces are covered with this epidermis. Okay, on the upper side it is simply termed as upper epidermis, and on the lower side it is termed as lower epidermis. Now these cell, if you see the structure of the cell, you can see the barrel shaped cells covered with a thick wax cuticle. Not only that, the lower epidermis actually consists of, you can see here, kidney shaped cells, right? Now these kidney shaped cells are actually guarding a small opening called as the stomata. That is why the kidney shaped cells are called as guard cells also, right? Guard cells help in opening and closing of this small pore called stomata, for exchange of gases mainly. Okay, right. So that is how the epidermis is seen. You can see around these guard cells there are the subsidiary cells, which are epidermal barrel shaped cells. Okay, right. So that is a surface tissue that actually combats with the surroundings, okay, surrounding environments. Right, let us see the simple permanent tissues and their types. Simple permanent tissues are, of course, you know now, made up of same type of cells, okay, only one type of cells. There are three main type of simple permanent tissues, which are the parenchyma, the collenchyma, and you can see the sclerenchyma over here, right? The parenchyma, you can see, are irregularly shaped cells. Of course, they contain many intercellular spaces, that is spaces between the cells. You can see the spaces between the cells, right? Okay, they are the most simple type of the permanent tissues, right? And they fill almost all the space in the plant body. Whereas collenchyma tissue is slightly rigid, gives mechanical support to the plant because they contain thickened cell walls, but at the corners of the cells. Okay, you can see here the cell wall is thickened only at this corner of the cell. You can see this corner of the cell, right? So the corners of the cell are being thickened, and such thickened cells are called as collenchyma cells. They give mechanical support to the plant. Whereas the third type is sclerenchyma tissue. Sclerenchyma refers to the fibers, long dead cells which have very thick cell wall made up of lignin and materials like suberin also. Now these long tapering cells actually are dead cells. They do not contain any cytoplasm, no nucleus at all. They contain only cell wall. Okay, these are the long fiber like tissue and they give mechanical support to the plant. Along with these tissues, there is another type of sclerenchyma called as sclerites or stone cells, which are found in certain parts of the plant. For example, the coconut shell, the nut shell. Okay, generally the nuts, when they have their shells, all those are made up of the sclerites, the stone cells, right? They are also dead cells and they have very thickened cell wall made up of lignin and suberin cell wall materials, right? But according to the certain functions, the parenchyma tissue redifferentiates into certain other subtypes. Okay, for example, the Sclerenchyma tissue. Sclerenchyma tissue, we learned last class that they contain many chloroplasts. That is why they seem to be green in color. Mainly, they are found in the leaves. Okay, the palisade parenchyma region and the spongy parenchyma tissue. All these are mainly the Sclerenchyma tissues, whereas they also form arenchyma tissue when they form air cavities in between them, large air cavities that gives buoyancy to the aquatic plants and help them to float on the plant. Okay, so that is how, and of course gives mechanical support over there. So these arenchyma tissues and the Sclerenchyma tissues are the subtypes of the simple parenchyma tissues, right? Now, let us talk about the complex permanent tissues, which we saw yesterday in detail. There are two types of these vascular tissues, which are the basic conducting tissue systems. The vascular tissues are of two types. One is the xylem and the other one is the phloem. Now, the xylem are water conducting tissues and the phloem are food conducting tissues. Remember, water is conducted from the roots to the other parts of the plant, whereas food is being transported from the leaves that undergo photosynthesis process to produce food. Okay, so food is conducted from the leaves to all the other parts of the plant for energy. Now you can see here there are 4 different types of xylem tissues. Okay, we know now that the xylem is a complex type of tissue. So how many types of cells it is made up of, tissues is made up of? 1, 2, 3 and 4 types. So the tracheids, the xylem tracheids, the xylem vessels, the xylem fibers and the xylem parenchyma together form the xylem tissue. Okay, whereas you can see here the phloem cells are actually made up of 4 different types of tissues. Here you can see the sieve tubes, the companion cells. Okay, I'm sorry. The companion cells, the phloem parenchyma and the phloem fibers. All these are living tissue except the phloem fibers, which are dead cells. Okay, right. So these are the complex permanent tissues we learnt in detail in the last session. We also saw how the plant tissues are arranged in a stem, a monocot stem and a dicot stem, where you can see the epidermis, which is the surface tissue, the ground tissue, and you can see the vascular bundles, that is the vascular tissues made up of xylem and phloem, right? We call them as vascular bundles. Okay, of course you can see in the monocot stem you don't find the cambium tissue here, but you can find that in the dicot stem. Now in the dicot stem you can see the surface tissue, which is epidermis, having some certain epidermal hairs sometimes, and the ground tissue which is made up of collenchyma and collenchyma and the parenchyma tissues. You can also see the vascular bundles made up of the xylem towards the inside, towards the center of the stem, the phloem towards outside the outer side of the, facing the outer side, whereas the cambium tissue, which is the, do you remember, lateral meristematic tissue, which is the cambium, is found in between the xylem and phloem in a dicot plant, that helps in helps in horizontal growth, or thickness or the girth of the plant increases because of this, right? You can see they are arranged in concentric ring in dicot stem, but in monocot stem they are scattered, right? So this is how plant tissues are arranged. In a leaf, you can see a green colored leaf, right? Because of the presence of the Sclerenchyma tissue in the palisade and the spongy mesophyll region of the leaf, that is the central part of the leaf. Of course, at the center of the leaf blade, you can see these the midrib and the veins, isn't it? You can see these veins here, right? Now all these are made up of the conducting tissues, the xylem and the phloem. Okay, now of course both these vascular tissues as well as the ground tissue is covered by the epidermis, upper epidermis and the lower epidermis. Understood? So this is how the plant tissues are arranged in leaves. So this is a schematic diagram showing different types of plant tissues which we just briefed now. But now just think about this question. Why are we talking about the tissues? Why we need to learn about tissues? We learned about animal tissues, we learned about plant tissues, but are they related to what is the topic of this chapter? Introduction to biotechnology. What is biotechnology? Why are we talking about tissues in biotechnology? Because we know that tissues and cells we learn under a different field of science called cell biology or cytology, right? Cytos means cell and logos means study or discourse. But why are we talking about the cells and the tissues and their structure and their function in this particular chapter? Of course, there must be some relation, isn't it? Right. So their location, their structure, their function, we need to learn about these tissues, because next we are going to learn something which is the very basic, which is a requirement of this field, a new field called as biotechnology. Okay, right. And that is how we are going to start with. In an organism, some cells, now we are talking about cells, right, are totipotent. Toti refers to total, potent refers to the potential or the capacity of the cell. But what kind of capacity? Okay, let us see. Few cells have a potential to produce a whole organism if provided a proper environment. But how can it produce? The property of the cells that can help produce the whole organism actually actually undergoes a long process that has led to the rise of a new branch of biology called as biotechnology. Here we require these cells, we require the study of cells, we require the study of tissues, we require the knowledge of the structure, the functions, as well as the location of those tissues, whether in plants or in animals. Okay, so here we are going to use this property of the cells called totipotent property, and to produce a whole organism. So a single cell can produce a whole organism. Yes, it can. Okay, and we are going to learn all these things under this field called as biotechnology, right? Now, with the help of this technology, it has become possible to produce improved varieties of plants and animals. You have heard about hybrids, isn't it? Not only hybrids, have you heard about clones? Yes, you have. Okay, so all these techniques actually are being studied under this field called biotechnology, which we actually use for human welfare. right? So it was realized that with the help of this property, which property? The totipotent property of the cells and their genetically controlled biochemical processes, varieties of high yielding crops and new species of plants, animals, and various vaccines could be produced. This has led to the rise of a new branch in biology, and we term it as biotechnology. Okay, so what exactly is biotechnology? Let us see. The techniques of bringing about improvements in living organisms by artificial genetic changes and by hybridization for the welfare of the human beings are together called as biotechnology. Okay, so this technology includes two basic techniques. One is the genetic engineering, and the other one is tissue culture, right? So we are going to study these two techniques in brief, of course. Details you will be learning in higher classes, but let us see what exactly these techniques are and how they help us produce new varieties and new hybrids of plants and animals, which we are actually consuming now. Okay, right. So what are the major uses of biotechnology? Let us see. It is used in the production of cash crops. Improvement helps in the improvement of varieties of these cash crops. It helps in increasing the abilities of plants to withstand environmental stress, right? Helps in production of vaccines. So vaccine production is also a very important use. Not only that, congenital diseases. The congenital diseases or the congenital diseases are being diagnosed as early as possible, and they could be treated well off. The next use of biotechnology is in organ transplant. You must have heard about kidney transplant, heart transplant nowadays, isn't it? This was possible only because of the field biotechnology. Not only that, nowadays even cancer research is based on this field of science, whereas production of artificial skin, cartilage, all these things in laboratories in the closed and controlled conditions can be produced. The production of artificial skin, artificial cartilage is being taken up in laboratories, of course, large laboratories, and all these are because of the field of biotechnology. Let us see what is biotechnology first. The techniques of bringing about improvements in living organisms by artificial genetic changes and by hybridization for the welfare of human beings are together called as biotechnology. Now, biotechnology has two basic process or two basic methods. One is the tissue culture, the other one is the genetic engineering. Okay, right. Let us talk about the tissue culture first. Right, so be prepared for an exciting and recent techniques of biotechnology. Okay, now the ex vivo growth of cells or tissues in an aseptic and nutrient rich medium is called as tissue culture. What is this tissue culture? And how is it going to, how do we perform tissue culture to produce new organism? So for that we need the knowledge of tissues, their structure, their location, their function, so that we can culture them. But where? In closed conditions, that is in laboratories. Okay, we can culture the tissue in such a way that they can produce a new whole organism. If it is plant, of course we call it as plant tissue culture. If it is going to be animals, we are going to culture animals, then we call it as animal tissue culture. So what is ex vivo growth of cells? Ex vivo refers to exterior of a living organism. When the cells can be grown in controlled condition in the labs, then we term it as ex vivo because they are outside a living system, outside an organism. They are being separated from the organism and grown separately. Okay, not inside the living organism but outside, but still they are cultured. Why they are called cultured? Because they are allowed to multiply, they are allowed to grow, right? So we call it as culture. Now here ex vivo growth of the cells or the tissues in an aseptic and nutrient rich medium. Aseptic, A refers to absence and septic refers to the germs. Now aseptic refers to germ free conditions. Okay, so we need to grow the cells ex vivo but in a germ free condition. Not only that, it also requires nutrients. Of course, every organism requires nutrient for energy, so nutrient rich medium is being provided to that particular tissue or that particular group of cells. Okay, and when we grow the cells in an aseptic and a nutrient rich medium that attains, of course, ex vivo growth, then we call it as tissue culture. Okay, with the help of tissue culture technique, a complete organism can be developed from a single cell or even from a group of cells. We term it as tissues, right? Okay, now a liquid, solid or gel like nutrient medium can be prepared from agar. Now agar is a substance that is being extracted from the algae, mainly the red algae, right? And this agar is, of course, is a gelatinous substance, is a gel-like substance. Okay, so using this agar, either a solid medium, nutrient medium, or a liquid nutrient medium, or a gel-like nutrient medium is being produced. We simply call it as agar medium or agar nutrient medium. Okay, this is a general process we are going to see, and generally we use agar in the nutrient medium. Along with, of course, supply of nutrients, various nutrients that are required for the normal growth of the cells is being provided in the nutrient medium, and the energy necessary for the tissue culture is used in this technique. Now, of course, every cell requires certain light, certain temperature, the aseptic conditions and nutrients to grow, right? So all this is being provided in the controlled conditions in the laboratory, and we can culture, we can grow the tissues and develop a whole organism by this procedure. So when we prepare for tissue culture, the most important is the agar nutrient medium. Okay, of course, different plants require different constituents, different nutrients, different proportions of different nutrients. Okay, so all these things will be standardized. Okay, and still the agar medium will be required. Well, let us see how this tissue culture is being done. Suppose we take a plant part, say this is a leaf. Okay, it's a leaf, and we are going to take a small part or a small portion of the tissues from this leaf. We term it as explant. X is removal. We are going to remove the small part of that plant part. Okay, now when we can take it, and the explant is actually inoculated in a nutrient medium. Now this is a petri dish. You can see a group of tissues are being formed. Small explants show growth because of the nutrient rich medium and the aseptic conditions and all the other physical conditions which are provided in the lab. These explants start showing irregularly growing cells. Now can you see, this is called as a callus. So development of callus takes place takes place in the agar medium, nutrient medium, and when this callus is being allowed to grow and multiply and form the new organism, that is a plant. Can you see a small plantlet here, a small sapling here, showing the leaf-like structure and stem-like structure and a root-like structure over here? Yes, this is the new plant, the plantlet that is being produced, that is being developed through this callus, okay, from a small portion of the leaf. Got it, all of you? Now this is how the basic tissue culture technique is. Now this callus, of course, can be separated, can be segregated. Even single cell can be segregated, can be separated from this callus and allowed to grow in the nutrient agar medium, and each cell can grow into a new organism. You can see here the small plantlets have already been developed in the nutrient agar medium, isn't it? Right. Now, but how did all this happen? Who discovered it? Who discovered the totipotency of the tissues and of the cells? Okay, it was this person, Frederick Campion Stewart, who is a British botanist, and he showed that the cells and the tissues can be grown ex vivo outside a living system, outside an organism. What did he do? He actually tried out isolating the cells from carrot root and cultured them in a nutrient medium, okay, and proved that each cell has the ability to produce the whole new plant. So he is the person behind this miracle, isn't it? The discovery of the totipotency of tissues and cells, right, was being done by Frederick Campion Stewart. Right, let us see how tissue culture in plants takes place. Just now we saw in brief how the plant part can be developed into a new plant, but there is a process, a procedure to be followed. Okay, now let us see. When a small tissue sample of the leaf is being taken from it has been scraped, just a small bit of the tissue of this leaf, suppose, is taken, it is being inoculated, introduced into the agar medium. So these tissue samples, either one or few, are being placed, okay, slightly embedded in the agar growth medium, agar nutrient medium. Now this agar medium contains all the nutrients for growth of the cells. So cells start growing and they form lumps of irregularly growth cells. Now these cells are called as calluses or callus. The callus can be separated and each and every cell can be segregated and introduced again into a new nutrient medium. It can be planted, and along with the nutrient agar medium, what is being added? Auxins are being added further. What are these auxins? They are growth hormones. Along with auxins are the main growth hormone, along with other two, that is the cytokinins and the gibberellins, together in specific proportions. They are being added one by one together or turn by turn, and that helps the samples, these calluses, to develop into tiny plantlets. Can you see small plantlets are being developed in the petri dish, in a small glass dish, okay, that was kept in the, of course, in the lab conditions, in the controlled conditions. Now these tiny plantlets, which are actually which have grown in the controlled condition, in the lab, and of course in the nutrient medium, they have never been exposed to the outer surroundings, to the outer environment. Now these plantlets, tiny plantlets, will be slowly planted in the compost, okay, and they will be allowed to get acclimatized, okay, of the surrounding environment, of the light, of the temperature, of the heat, and of the soil, of the organisms present in the soil, the organisms present on the land. All these, they will have to face all these things, and still they withstand all these upcomings and start growing well in the soil. Now that part is simply termed as hardening. So we are going to see how exactly the various processes takes place in tissue culture. Now suppose for example we take a banana plant. Okay, this is simply called as the source plant. Now the source plant, we have chosen the bulb or the comb, okay, as this from the source plant as the explant here, right? And it is being introduced, a small part of this explant of this bulb is being bulb comb is being introduced into the agar nutrient medium, right? Yes, here we are using a bottle, right? A glass bottle, anything which is aseptic and will not interfere in the growth of the tissue can be used. Okay, so this is the primary treatment where the explant, the part which we are going to remove from the source plant, will undergo certain treatment, primary treatment. Of course, the aseptic condition has to be followed there, otherwise whatever materials you are using, whatever tissue samples you use, may have to be discarded otherwise, because microorganisms contaminate the whole technique. Okay, so we have to be very careful about the aseptic condition, and that has to be followed. How do we follow? Generally, the agar nutrient medium is being autoclaved, that is, it undergoes a certain amount of, it is being heated, boiled in under pressure, okay, in certain devices called autoclaves. Now where every other microorganism is considered to be to die and to disintegrate. Okay, so this particular procedure simply is being termed as sterilization procedure. So the aseptic conditions can be maintained in the lab using certain sterilization procedures, of course using certain chemical vapors where or UV light rays, where that particular region where we are going to inoculate the explant in the nutrient agar medium, in the germ-free nutrient agar medium, will be made aseptic. Okay, and so as no microorganisms need to contaminate this particular environment in which the explant is being inoculated. Okay, now the explant is left there in the lab under certain amount of light, of course a certain optimum temperature for growth, and is left to grow and multiply. So after the primary treatment, the explants, the tissues start multiplying, starts growing and get converted into callus. Now callus is being transferred into another setup containing the nutrient agar medium with plant hormones that initiate the development of shoot system and the root system, okay, in definite proportions. When the growth hormones are added in definite proportions, of course the shoot and the root starts growing, so and the cells start multiplying rapidly. Now, so you can see here the shooting and the rooting that has taken place in these bottle jars, okay, right, which are being used for tissue culture. Now, after the rooting and shooting is being done, slowly the plantlets which are being grown inside the control conditions, inside the labs, has to be taken outside in the natural environment, and they have to be hardened. That is, they will have to face all the conditions of present in the environment, the natural conditions. So they have to get used to it or acclimatize to the surroundings and the soil, right? Surrounding environment in which it has to undergo, it has to face the temperatures, the wind, the water, the rain, the temperatures, the um, humidity, so humid conditions, all these different kind of conditions in the prevailing in the environment, natural environment, has to be provided to these plantlets so that they become hardened, that is they become capable of developing and growing in the natural environment. So this part is a hardening technique that is called as initially the primary hardening technique and the secondary hardening techniques are being undertaken. Now initially the primary hardening technique is to bring the plantlets into the soil. Initially the compost is being used, slowly mixed with soil, and these plantlets are brought outside in the environment for few hours in a few hours, so that they can get used to the light, used to the temperature, used to the natural conditions. Slowly they are being transferred, okay, into the pots, right? Flower pots, small or large flower pots, and slowly into the fields. They are being transferred into the fields, into the natural environment, where they can start growing profusely and be successful, even though they are being cultured. They are the cultured plants. Okay, so in this way, in this manner, using various processes and various techniques in the tissue culture, we can develop a whole organism from a small part of that particular organism, particular small tissue of that part of that organism. So source plant can be source of so many plants, isn't it? And if at least 50% of, if we consider 50% of these plantlets survive the hardening procedure, then the whole earth will be, of course, green, isn't it? Right. So let us see. You can see here how the plants are being developed, and by the tissue culture technique, of course, and farming is being done. You can see the banana plants here in greenhouses, how they have been developed in the greenhouses here, undergoing the hardening process, and then in the natural environment they are just growing lushly, isn't it? Right, profusely. And that is the beauty of this technique, the biotechnological techniques. Okay, tissue culture. Right, next let us just see the second method of biotechnology, which is genetic engineering. Now regarding genetic engineering, let me tell you, you'll be studying in brief and in detail in higher classes, but let me just introduce you to this field. Genetic engineering, actually the deliberate modification of the characteristics of an organism by simply manipulating, changing its genetic material. Which is a genetic material? We know that it is DNA for all the organisms except viruses, right? Except few viruses, I would say. Right. So the DNA, there is a change that is being made to manipulate the DNA itself, and because DNA is the main genetic material of every organism that store the information of all the characteristics of an organism, if it is being manipulated, is going to produce the desired characteristics, okay, in the new organism. And that is how, of course, the small change in a small part of the DNA, which we call it as a fragment of DNA, called as gene. If even a single gene is being turned over or is being manipulated, then the whole organism may show some or the other different characteristic, okay, other than the natural one. So that process is genetic engineering. Let us see the process of genetic engineering just in short. Okay, now genetic engineer, this is not tissue culture, this is genetic engineering, where we use certain microorganism as experimental vectors help to transfer the genetic material from one to the other cell, okay, and other parts of the plant or of the culturing explants which are being cultured. Now if you see here the potted plant, a small part is being used as an explant. The leaf explants are used, which are being actually introduced into a nutrient agar medium along with an agrobacterium, which is the experimental bacteria. What this bacteria does is it actually enters into the tissue, into these cells, into the cells and the tissues of the explants, and will start helping the explant produce manipulated cells, manipulated with the genes, okay, right. And of course the tissue starts developing here in the agar medium and produces the irregularly growing cells. We call that as callus, right? Now these calluses actually are going to develop into new plants. Of course, in the way that plants grow in the tissue culture in the lab, the same way this can also be grown. But now these plants are not normal natural plants. We call these plants as the genetically modified plants, or the GM plants, genetically modified plants, or simply termed as transgenic plants, in which the genes are being transferred, the genes are being manipulated. Okay, now that is how the genetic engineering takes place. This is one simple method, the basic method which I've told you. You will be learning different methods under this method of biotechnology in higher classes, but that is going to be very interesting. Right, let us see what are the changes in agricultural management due to biotechnology that has taken place. Now, we just now learned how the genetically modified crops are being developed, okay, by introducing changes in the DNA of natural crops and other plants. These GM plants, or the transgenic plants, exhibit the desired characteristics like to withstand the environmental stress, the insect resistant, the pest resistant, the chemical resistant, that is the chemical fertilizer resistant, the drought resistant, the weedicide resistant, okay. All such varieties can be produced, can be developed using these genetically modified, the biotechnology process called genetic engineering. Right, better quality seeds, high yielding varieties of GM crops like the banana, the maize, the rice, cotton, potatoes, corn, all these different types of plants can be produced with better quality and good quantity. Now, due to the use of seeds of the GM crops, there is improvement in nutritive value. See, look at this, and decrease in the loss of crops, which is very, very important nowadays because we need lots of food supply, isn't it? As the population of human beings is rising higher and higher. In this way, as seeds of better quality of crops are being produced, farmers all over the world are cultivating the GM crops, of course, on large scales. The high yielding varieties of GM crops like banana, maize, rice, potato, soya bean, tomatoes, cotton, apple, brinjal, papaya, beet, rose, my gosh, so many different types of genetically modified plants are available nowadays. GM crops are available nowadays, and we are consuming the food Is that clear? Now, let us see the pest resistant genes have been introduced in some of these GM crops also. For example, you can see the maize Mon 8110 and Mon 863 are the two varieties of the maize where the pest resistant genes are being introduced. So they are pest resistant varieties. The potato, the name of the variety is Amphora. And the golden rice, can you see the golden rice, which is being developed for vitamin A content? Okay, you are going to learn this about this in the higher classes in detail. Anyway, the cotton, the BT cotton, now this is another interesting GM crop. Which is simply called as BT cotton. Okay, you can see a picture of even tomatoes termed as Vaishali. Nowadays, most of us must be eating these tomatoes, a large and tasty thick tomato with thick pulp, right? Anyway, now in this way, actually, now a green revolution, should I say green evolution, is being achieved through tissue culture. And a country like India, of course, with a huge population, we are actually meeting with success in producing sufficient food grains for all nowadays. So all this is possible only because of biotechnology, that when we made changes in agricultural management, we are able to meet with success and provide food for all, right? Now let us see what are the applications of biotechnology. Applications of biotechnology can be seen in various fields like floriculture, nurseries and forestry also. Nowadays, there's a lot of land that has become barren, isn't it? Because of deforestation, because of other reasons, because of changing climatic conditions. So all these conditions can be revived again using biotechnology. We can apply techniques of biotechnology to make our earth greener and greener again, right? So nurseries, which are necessary for various purpose, like growing gardens on a large scale, afforestation as well as reforestation purpose. Seedlings have to be supplied in large numbers for all these purposes. Of course, we can supply that using tissue culture techniques. It is profitable in such cases to produce plantlets with the help of tissue culture techniques. And you can see here a small picture of the nursery where, of course, you can get any kind of plant, right? The tissue culture can also be used to grow these plants on large scale, which can bear flowers. Nowadays, you get beautiful hybrid flowers, beautifully colored and colorful flowers, okay, which we have not even dreamt of. Okay, now even the fruits, different hybrid fruits, hybrid varieties, high yielding varieties, large sized fruits we get, isn't it? And all these are of excellent variety, and we have attained all these, man has attained all these because of the tissue culture techniques, okay, and of course the genetic engineering methods. The fully grown plants can be produced in shorter durations. Nowadays, a special device called as a bioreactor are being used where cells can be grown in more large amount of nutrient medium. So large number of cells can be grown, and they can be protected from pathogens, that is from the harmful microorganisms. So damage will be very negligible. Okay, the bioreactors are actually useful for producing plantlets on a very large scale. So you can see a large bioreactor here, can you see? And of course you can see that the genetically modified crop varieties can be produced in large quantities, okay, in bioreactors. Nowadays, this is being carried out in various parts of the world. Now, plants can be grown on large scale even if means of pollination or germinating seeds are not available. Look, say we need not require the pollinating vectors. We need not require the seeds that has to wait and germinate and take long years to produce the plants, right, and the large trees. For example, we can also produce varieties of orchids and pitcher plant. Can you see a small picture of the pitcher plant is being shown, the orchids, beautiful flowers, right? They do not germinate, but these plants can be they do not germinate easily in any kind of environmental conditions, but the plants can be produced by means of tissue culture. Large number of seedlings, plantlets can be produced in short time, okay, using minimum resources and minimum materials. That is also a very important application. So less material and less time, but large number of seedlings can be produced. Usually, plants produced by tissue culture and genetic modification techniques actually are disease-free plantlets. Okay, now these plants, plants which are actually developed by tissue culture of meristems. We have learned meristems, isn't it? The apical meristem, the intercalary meristem, and the lateral meristem. If we are going to produce the plantlets from the meristems, then these plants will be even virus-free plants. So even viral diseases will not occur in these plants. Okay. Another application, if you see the embryos that are formed by conventional hybridization technique between two or more varieties may not fully grow for some reasons. Sometimes it so happens that the hybridization technique goes wrong and the hybrid variety cannot be produced properly. In that case, the embryos which are being produced by the tissue culture technique always complete their full growth. So this is also very, very important. Now, the application of biotechnology can also help in conserving the rare and endangered plants which can be grown by simple tissue culture technique and can thus be protected from extinction. If we would have known about this technique few years earlier than we discovered it, it would have been better, isn't it? So we could have saved many plants, we could have saved many animals also from extinction. Similarly, various parts of and seeds of such plants and plantlets can be preserved by tissue culture, and of course those varieties can also be protected, right? So these are various applications of biotechnology. Now we come to the end of this session. Let us see what did we learn in this session. We learned about what is biotechnology, the methods of biotechnology, that is tissue culture, and the genetic engineering methods, which are the basic biotechnological methods, right? I hope you must have understood these methods, maybe in brief, but still it must be very clear. Now, changes in the agricultural management due to biotechnology, as well as the various applications of biotechnology in various fields like floriculture, nurseries, and forestry. Thank you all for joining me in this session, and keep good health always. Be fit and fine. Have a good day. Thank you, all of you. Bye.